Hello, everyone. Before I start, thanks for the RECOMB ISCB conference for allowing me to have this talk. The topic that I'm going to talk about today is deep integration and optimal experiment design in multi-omics genome scale models. So what is genome scale model? Genome scale model is the model that predicts cell state and cell behavior from environmental settings and genetic background of the cell. The example of cellular state include, for example, molecular expression level, and example of cellular behavior include growth dynamics for bacterial cell. And for example, for environmental settings, oxygen level or chemical composition of medium. And the accurate prediction from genome scale model will make broad impacts in biomedical sciences. For example, in synthetic biology, we often design the gene circuit, and we would like to know what are the best environmental settings and host cell for the gene circuit to function. And in metabolic engineering, we synthesize metabolic pathways on host genome, and we would like to know what the environmental settings and host cell would maximize the yield of engineered product. And even in predictive medicine, for example, we have more than 100 existing antibiotics, and this antibiotic effect can be stronger by combining multiple antibiotics. And the question is how we are going to know what would be the best combination without spending too much experimental cost and time. And all these questions can be answered by computational simulation of genome scale model. And even genome scale model can guide us to what experimentation we need to do by finding the condition with the most uncertainty. But there are many challenges to achieve this goal, which we can categorize them into three groups. First is data challenge. Often we have noisy, inconsistent, and metadata lacking data sets once we combine all the different data sources from public available repositories, and which that makes it harder to build the accurate model. And also we have modeling challenge. For example, we have often incomplete knowledge for an organism. For example, E. coli, although E. coli has been known that is one of the most well-characterized organisms, we still have incomplete knowledge for that organism. And also model complexity is so high because there are so many biological parameters to fit. And the training and testing of such model is tricky and inefficient because, again, it's because the model complexity is so high. And we have third challenge, which is how we are going to refine this model, how we are going to select a new experiment for improving this model. And we shouldn't rely on the biased or intuition-based or lab-centered strategy. And there has been many tremendous efforts to tackle these challenges. For example, we have, for data challenge, we have very interesting compendium out there, for example, Columbus or Moped, that integrates the transcriptome or proteome, for example. But we still need to advance ourselves to integrate the more comprehensive data that includes for example, the metabolome fluxome as well as phenome data. And once we build that, we have also, in previous years, we have many interesting genome scale models out there, but still we need to improve our model strategy that is trained based on the full layered information to represent the most comprehensive knowledge of an organism. And for the challenge, which is for model refinement, we need to have the systemic guidance for omics profiling to improve genome scale model, although there has been many studies highlighting this issue. So here, we propose to build the normalized full-layered multi-omics compendium that ranges from the transcriptome, proteome, metabolome, fluxome, as well as phenome data. And once we build that, based on that research, we build a genome scale model that integrate all that layers. And after we've done that, we go into the how we are going to add a new experiment for refining the model based on optimal experiment design, specifically designed for omics profiling to improve the model performance. So here we have built the multi-omics compendium, metadata annotated compendium for E. coli that ranges from transcriptome, proteome, fluxome, metabolome, and as well as phenome. And also we did tremendous effort to characterize each condition of those profiles. For example, we annotate what genotype of strains that they use and what stressor that they use, and also what chemical composition, for example, sugar or the salt sources in chemical composition medium, for example. And also to connect the different molecules in a model, we also integrate all the network information from publicly available repositories or literature, for example, metabolic reaction network, signal transduction network, protein-protein interaction network, and transcriptional laboratory network. So we combine all these research into one repository to guide the model and as well as the community to use that research depending on what they would like to use. So once we build this research, how we are going to build the model? So we're starting from the input layer that has more than 6 features that consist of strain genotypes, genetic perturbation, and stresses exposed, and chemical composition of medium. Once we characterize the input layer, then this information goes into the transcriptome layer that predicts transcription expression levels on genome scale using recurrent neural network. And then this information again goes into the proteome layer that predicts protein expression levels on genome scale using 4 different network information. And then we also go to fluxome layer and metabolome layer to predict metabolic fluxes and concentration of metabolites from input transcriptome proteome layers. And finally, we go into the growth dynamics, in phenome layer to predict the growth dynamics by combining all the information from the 5 different layers. So we have validated this method layer by layer. For example, for transcriptome layer, we have shown that the performance is better than 3 different baselines. And also this performance is getting increased when we focus on transcriptome factors only. And for proteome layer, we were able to see that the performance based on the consensus of 4 different methods was better than any of single method, as well as the 2 different baselines. And this observation was also present in the other layers, for example, metabolome and fluxome, when we compare to the other method and baselines. And the final layer, which is the phenome layer, we mainly focus on growth rate prediction. And the way we predict growth rate is as follows. So we predict the growth rate based on consensus prediction from 5 different layers. What I mean by that is we predict growth rate from input layer, transcriptome, proteome, metabolome, fluxome. We combine these predictions to make final growth rate prediction. And that consensus prediction was better than any of single layer. And that consensus prediction was better than when we combine the less layer information, meaning that, for example, it is better than the consensus prediction that integrates only 4 layers, for example, input, transcriptome, proteome, metabolome, without fluxome layer. And we were able to see that this prediction performance was getting up when we focused on novel wild types up to Pearson correlation coefficient 0.76 from Pearson correlation coefficient 0.65. And we next ask how we are going to improve this model, how we are going to decide what experiments we need to do. In order to answer that question, we devised a remove method, and the way it works as follows. So first, we identify unexplored condition in the compendium, and then we next find the condition that maximizes entropy and prediction interval. In other words, we find the condition with the highest uncertainty, and then we do the RNA-seq profiling for that condition. And then we included data with normalization in the compendium, and we trained the model for improving the model performance. So as a proof of concept, we focus on the space of 85 different conditions, where each condition is a combination of 7 different carbon sources and 14 different stresses. Optimal experiment design that we devised was able to select a series of 15 optimal conditions. In this plot, the dark blue dot represent the condition that is selected by optimal experiment design, and the number represent the order. So the optimal experiment design tells us that we need to first experiment this condition, which is the butanol stress and galactose carbon source, and then go into second one, which is the butanol stress and glucose 0.2%. We go and go again until we reach to 15 condition, which is one of the example of the antibiotics and galactose. And one of the interesting observations from this figure was that some of the stresses are never picked from these 15 optimal conditions. For example, CFS, cold MCM. By the way, CFS and MCM is the only example of antibiotics. And we were able to understand why that happens by looking at the scatter plot between entropy and prediction interval and before and after refinement. And the entropy and prediction interval of 3 different stresses are actually getting down after refining from the profiling of different stresses. In other words, the profiling of stress A was actually decreasing the uncertainty for the stress B, for example, because stress A and B are related to each other. So we validate this optimal experiment design by really profiling the RNA-seq for 15 optimal conditions that we selected. And we were able to see that prediction interval was actually getting down when we add more optimal conditions. And also we showed that the prediction performance of the genome-scale model actually going up when we after refinement of 15 optimal conditions. So in summary, we have built the full-layered multi-omics compendium of E. coli, which we called RECOMB, which is most comprehensive multi-omics compendium for an organism. And we are preparing for the online accessibility for this compendium, but we're still under progress. And we have shown that we built the first data-driven genome-scale model that integrates 5 different omics layers. We also shown that this is a first application of the optimal experiment design based on genome-scale, based on the omics profiling for improving genome-scale model. And right now we are working toward tremendous effort to find new biological discoveries using this model. And finally, I'd like to thank my advisor, Elias Takobos, and I'd like to thank my lab members who participated in this project. Thank you.
Thanks for a great talk. One of the questions I have, and it's not really clear to me, is how do you choose, which layer to start with?
Right.
Because they all kind of like interact.
Right. Very good question. Actually, we designed this which layer need to start first is based on the utilization of the research that we have. So the reason why we go to transcriptome layer first is because we have a lot of data for transcriptome layer. And we wanted to first start from there because there are a lot of data again. Yeah.
I would argue that actually the most known layer is the metabolic one.
So when we talk about the metabolic layer, it means that the concentration of the metabolite. Right. And if you look at the figure that I made for this, the multi-omics compendium that has all the data points, right? So if you see here, our research number of profiles for metabolic layer is only around 700, but transcriptome layer we have more than 3,500 profiles. So we're starting from because we have a lot of resources for that layer.
Thanks.
Hi, great talk. Can your model make predictions about what happens when you perturb individual molecules somewhere else other than the input layer?
Right. So as I mentioned, we use the recurrent neural network for the predicting the transcriptome layer, but yes, I didn't mention about how I use, what kind of method I use, but here we primarily use the lasso base to select which features are informative to predict the predictor variable.
Great talk. Can you comment on the optimal experimental design? So was it a sequential design based on Gaussian processes, or?
Yes.
So it was sequential, and you fixed the number of potential experiments.
Right. So we actually use two different uncertainty, and one is the entropy and the other one is the prediction interval. And prediction interval was measured from the recurrent neural network, and 1,000 recurrent neural network to get the interval, right? And for getting the entropy, we use the Gaussian process. And Gaussian process, the reason why we use the Gaussian process is because we wanted to model the joint distribution between all the conditions that we have. So that's why we use the Gaussian process for that.